Thank you, and I appreciate the opportunity to present some of the work that we're doing in COVID-19 and the respiratory tract microbiome. These are my disclosures. So SARS-CoV-2 initially replicates in the upper respiratory tract, and in mild disease, it remains localized there, or in severe disease, it propagates to the lower respiratory tract. And we know that the respiratory tract microbiome can affect outcome from viral respiratory infection through modulation of innate immune function, gene expression, or other mechanisms. And that's work that's been carried out to a limited extent with RSV, influenza, largely in animal models, but some suggestive human data. And we're interested in this topic because the oropharyngeal microbiome may impact not just upper respiratory tract processes, but it's also the source for the lung microbiome in people without significant lung disease. And so the connection between the oropharyngeal microbiome and the lung microbiome is significant. So we started out to ask the questions, what are the initial and longitudinal features of the upper and lower respiratory tract microbiome in COVID-19 patients? And are there features of the microbiome that are linked to disease severity and outcome? So we collected just over 500 longitudinal samples from 83 hospitalized subjects. And on the WHO scale, that means that they were all number 4 and above, because 4 is hospitalization. We collected oropharyngeal, nasopharyngeal, longitudinal samples, and on patients who are intubated, we collected endotracheal samples as well. We carried out 16S rRNA gene sequence tag sequencing for the bacterial microbiome. We did targeted qPCR for 2 small commensal DNA viruses of the airway. We compared COVID-19 to a dozen non-COVID ICU patients and a similar number of healthy controls. And we analyzed the microbiome in relationship to clinical outcomes on the WHO score of 0 to 10, 10 being fatal, 4 being hospitalized. And we also looked at the relationship to systemic immune responses. We also analyzed the SARS-CoV-2 levels in our various specimens. That data is shown here and has been published, but I'm not going to talk about that today. So the first thing that we saw is that the upper respiratory tract microbiome was associated not only with COVID-19 as a disease, but also with disease severity. So here I have principal coordinate analysis by unweighted unifrac of the oropharyngeal microbiome and the nasopharyngeal bacterial microbiome. And here we've taken in orange our COVID-19 patients and in blue healthy subjects. And we could see both the OP and the NP microbiomes are significantly different from healthy subjects. Now here we've taken our subjects and we've compared our COVID patients to our non-COVID critically ill patients. Not all of our COVID patients were critically ill, but we compared them to critically ill patients. And so in green are the non-COVID patients, and in the various shades of brown through pink are our COVID patients. And what we could see is that our oropharyngeal and nasopharyngeal microbiomes, our non-COVID patients, differ from our COVID patients. And then by Perelman testing, we can see that the microbiome communities differ according to the degree of ultimate disease severity that the patient reached. Each patient was categorized based upon the ultimate level that they reached. We also looked at individual taxa. Here we have at the phyla level in the oropharyngeal and in the nasopharyngeal communities. We see significant differences across our non-COVID and our different degrees of COVID severity, particularly in Bacteroidetes, Proteobacteria, and in both the oropharyngeal and nasopharyngeal. And then when we looked at specific genera, it typically was the loss of normal upper respiratory tract flora seen in the COVID patients. Well, we then asked, what are the longitudinal profiles of these patients? So the first thing that we asked is there any relationship between the first sample that we got on each patient and the ultimate level of disease severity that they reached over their entire course of hospitalization? And this panel on the left shows on the x-axis the maximum WHO score reached by a patient, and on the y-axis is the Simpson diversity in the initial sample that was taken on that patient. And there's a significant inverse relationship between diversity and sicker patients. So those patients that ultimately reached higher levels of severity, with 10 being fatal, had lower diversity. We also found that in longitudinal sampling over time, the oropharyngeal microbiome changes more rapidly in COVID-19 patients than in critically ill non-COVID patients. And here we measured the unifrac distance from every longitudinal sample from the initial sample in that patient, and we see that there's more rapid divergence over time in COVID in orange than non-COVID patients, suggesting that there's less stability of the oropharyngeal microbiome in COVID-19. We had a subset of patients who were intubated, and we had samples from 24 subjects. And on the right here, this shows by bar graphs the composition of the endotracheal microbiome. These are our 24 subjects. On the x-axis is days from intubation. Above the horizontal line, we can see what culture results the patient had. And below this horizontal line, we see the distribution of taxa in these endotracheal microbiome. On the left here, we can see the diversity in our COVID-19 endotracheal aspirates as compared to healthy subjects. So the first thing that we see is that there's a marked loss of diversity in endotracheal aspirates of COVID-19 patients compared to the bronchoalveolar lavage of healthy people. The second thing we see is that people can differ in terms of their stability in the endotracheal microbiome. So we have some subjects in whom we have relative stability over time. Here, this subject, and here this subject with a similar pattern. And then we have other subjects that show gradual change in the microbiome profiles over time, such as this subject and this subject, gradual evolution. And then we have other subjects in whom there are marked rapid shifts showing instability of the lung microbiome over time. We found that Staphylococcus was particularly dominant here, and 6 out of our 24 subjects were heavily dominated by Staphylococcus. You can see that in yellow. And we also saw an unexpected dominance of Enterococcus in quite a number of subjects. 3 subjects were dominated by Enterococcus. And the last thing we saw is that the sequencing data was only partially concordant with what was determined by culture. We next asked whether the oropharyngeal microbiome had any relationship to systemic immune responses. So the first thing we checked for validation was whether our WHO severity score correlated with the lymphocyte to neutrophil ratio. That's something that's been shown by many other groups. And indeed, the more severe the disease, the lower the neutrophil to the lymphocyte to neutrophil ratio. So that confirmed the expected pattern in our population. We then asked whether the lymphocyte to neutrophil ratio was associated with the oropharyngeal microbiome. And this middle panel shows that there's a significant relationship between diversity in the oropharyngeal microbiome on the x-axis and the lymphocyte to neutrophil ratio. So systemic immunity is also associated with the OP microbiome. Finally, we had a set of 34 subjects who were also co-enrolled in a study of deep immune profiling of PBMCs, in which a large number of individual immune parameters were defined, and then those were reduced in a UMAP distance matrix that was able to integrate them into a 2D matrix. So we compared the two-dimensional matrix of our immune profiles with the two-dimensional matrix of our microbiome profiles, and using two different statistical tests, Procruste's analysis, which is what's shown here, and a Mantel's test, we found a significant relationship between the communities in the oropharyngeal microbiome and the profiles of the subject's peripheral blood mononuclear cells. And here, the line connects the immune PBMC and the microbiome profiles of each subject, with the green triangle being the microbiome profile and the orange circle being the immune profile. So there was a significant relationship between microbiome and systemic immunity. We also looked at these two small circular viruses, anelloviruses, which are present in most or all humans, as well as redondo-viruses, which are a small circular DNA virus that we described recently in oral and respiratory tract specimens. And we found that both anelloviruses, shown here in orange, and redondo-viruses were significantly elevated or significantly more prevalent in patients who are intubated than in patients who were non-intubated. This shows the proportion in orange of intubated patients positive for anelloviruses, significantly higher than those non-intubated. Same thing for anelloviruses here in blue. This was by qPCR. Finally, we asked whether there are integrated features of the upper respiratory tract microbiome at early time points that associate with or can predict peak disease severity reached during the course of hospitalization. So we use random forest models to sort maximal WHO scores. And here we've broken it down by disease severity. And by random forest, we see an area under the curve for the oropharyngeal microbiome of 0.82, nasopharyngeal microbiome of 0.75, in demonstrating significant accuracy in predicting the maximum disease severity. And on the right, show the various taxa that are particularly predictive of those outcomes. We also found that the microbiome predicts intubation status with an even higher area under the curve, and the small circular viruses were the strongest predictors of intubation status. So our conclusions are that the upper respiratory tract microbiome in COVID-19 differs from both healthy and non-COVID patients. Early time point microbiome features associate with the ultimate level of disease severity over the course of illness, and they also associate with features of systemic immunity, and that the endotracheal microbiome in intubated patients is variable, can be highly unstable, and is frequently dominated by pathogens, particularly Staphylococcus. So we're particularly interested in this second point here, and just to unpack that a little bit, the key question here is really, is the microbiome findings a cause of COVID-19 severity, a contributor to severity, or is it simply a consequence of COVID-19 severity? And we're interested in this because we are interested in the question of whether a dysbiotic upper respiratory tract microbiome prior to SARS-CoV-2 infection might actually contribute to disease severity. Regulate whether infection is limited to the upper respiratory tract versus propagates to the lung, and the question of whether pre-infection upper respiratory microbiomes and the linked lung microbiome may contribute to the risk of COVID-19 severity in people with comorbidities through regulating inflammation, regulating ACE2 receptor expression, other mechanisms of regulating virus replication, as well as can the upper respiratory tract microbiome serve as a biomarker for predicting disease severity and outcome. And that's the area that we're working on right now. So let me end with acknowledgments. This work is done in collaboration with Rick Bushman, and the fantastic people who actually carried out the analytical work. I'll particularly mention Sam Whiteside, Guanzhong Liang, and Carter Merenstein, who did the analysis, and then also acknowledge our clinical team, who worked like crazy people all through the COVID-19 epidemic. I'll acknowledge our funders and several other collaborators here. The bulk of this work was just published recently and is available complete here in MBio. Thank you.